So we said they're weak acids. They can give away their hydrogen, and then what's left over, when they do that, is they form what's called a carboxylate ion. Okay, carboxylate ion. And this is going to feel confusing, but let me explain something. We name carboxylate ions with the name ending ATE. Okay, why is that confusing? What else do we name ATE? We just went over it. What functional? Yes, we name esters ATE. So why are we reusing this? That's confusing. But we use the word ion, okay? Carboxylate ion. So you will recognize this is different than an ester. It's not an ester because it will always have a charge associated with it because it will be an anion. Okay, if carboxylic acids give away a hydrogen, what's left over is an anion. Okay, so we'll name it that way. So if you have acetic acid, and here, can y'all see the yellow when I do yellow, or is that really hard to see? Y'all can see it? Okay. When they give that hydrogen away, what's left over is, we call it the acetate ion. So back in the day when you were introduced to polyatomic ions, if y'all even remember that, this was one of the polyatomic ions, okay? Acetic acid becomes the acetate ion. And when it gives that hydrogen away, somebody usually accepts it. It can be a base, it could be water. So for another example, if you have pyruvic acid, you don't have to know, I don't expect you to know what pyruvic acid is. But it is a carboxylic acid. When it gives its hydrogen ion away right here, we have the pyruvate ion. Now this is different than an ester. Why? Because you have this negative charge associated with it. So we put the word ion on there, okay? Now, if these anions are in the presence of like a group, main group 1 or main group 2 element, they form salts, okay? So like if we had like the acetate ion, you could have like potassium acetate or sodium acetate or lithium acetate or calcium acetate, okay? Those are commonly joined up with group 1 and 2 elements to form salts, okay? So for example, I'm just gonna write one of those out. CH3COO, that's the acetate anion minus. If you have Na plus, this would be sodium acetate, okay? This is the sodium cation part, this is the anion part. Usually when we write it in a compound though, you write the cation first. So it's kind of weird that I wrote the anion first, but so that's just an example. The reason I want to point this out is because in the body, this is how carboxylic acids exist at a pH of 7.4, which is physiological pH. They're not carboxylic acids, okay? They exist mainly as carboxylate anions. So we call them by their anion names, all right? That's how they are in the bodies. All right. Now we said they're weak acids, so they don't 100% dissociate, okay? So we have a certain concentration of acetic acid in the body. A portion of it will exist as the acetate anion, and a portion will not, and that all depends on Ka and pK values. Anybody just vaguely in the recesses of your mind remember Ka and pK? Like you've heard these terms before. Anybody just like, let's be honest, I've never heard these terms before. I need to know. Okay, so we need, this is all chapter 7, equilibrium, okay? This is quite a complex topic, and I'm not gonna cover the whole thing now, but Ka, there's an actual reaction that we derive Ka from. I'm not gonna do that here. That's a previous chemistry class. But Ka has a value, okay? And that value, all these, you don't have, I'm not gonna ask you to calculate Ka on exam. What I would do though is I would give you a Ka, and I would ask you based on the value of Ka. So we have all these Kas for different acids here. I might ask you to tell me which one of these acids is strong, or which one is stronger or strongest, and which one is weakest or weaker. Does that make sense? I would ask you to compare Ka values and judge the strength of the acid based on that. That's what I might do. So you need to understand a little bit about Ka. So basically, here's the trends you need to know. The smaller the Ka value, the weaker the acid is. So I want to explain something. We know these are reversible reactions, which is why we can get a Ka value. You can write a Ka for any reversible reaction. So the acid is the put together form. The anion is the dissociated form, all right? So the weaker the acid is, the less anion form you have, and the more it's put together in its acid form. Okay, the stronger the acid is, the more of the anion form you have. So if it's a weak, really the weakest acids, they favor the acid form, like the non-dissociated form. The stronger, notice I'm not calling these strong acids, I'm just saying they're stronger. They favor the dissociated anion form. Okay, that's about as much explaining as I'm going to do for acids. And how is this related to pK? Well, what is pK? Anybody remember what the function P does when you put it in front of a value, like pH? Here we have pK. Anybody?
Doesn't the pH value make it more acidic? Is that what you're saying?
On a pH scale, lower pH values are more acidic. Yes, Madison. But here, I'm just kind of wanting to define what pKa is mathematically. P stands for the negative log of something. Don't remember that? So like pH is the negative log of the concentration of H. We did a lot of that when we calculated stuff with acids and bases and calculating pH. So pKa is the same thing. It's just the negative log. You guys have a calculator with a log function. You might want to bring a calculator for exam 2, just in case. You can't use your cell phone as a calculator. It'd be the negative log of any Ka value, okay? Again, I'm not ever going to ask you to calculate a Ka value on the exam, but if I give you the Ka, you could calculate the pKa. Now, what's really nice about the pKa versus the Ka, on these scales, you can see that we kind of have these nice decimal numbers or whole numbers, and we don't have these crazy exponents. So how does pKa and Ka relate? How are these related? Well, the smaller the Ka, we said the weaker the acid, and it's opposite for pK. The larger the pKa, the weaker the acid. So let's look at this table, and we'll prove this to ourselves. At the very, very top, we have trichloroacetic acid. At the very, very bottom, we have glutaric acid. Okay. Now it gives you the structures, but I'm not interested in that right now. I want to look at these last two columns, Ka versus pK. I'm going to zoom in on trichloroacetic acid, the top one. Okay, what do you notice about the Ka value? 2.3 times 10 to the minus 1. Y'all know what 10 to the minus 1 means? Do we understand negative exponents? So like, which one is bigger, 10 to the minus 1 or 10 to the minus 6? Which one is bigger? A bigger number.
10 to the minus 1.
Yes, 10 to the minus 1, when you actually write the decimal out, is 0.23. 3.8 times 10 to the minus 6, if you write that out in decimal form, is 0.1234538. You'd have to move the decimal 6 places to the right to get it back to 3.8 times 10 to the minus 6. Okay, so if you're comparing these numbers, 0.23 to that number, obviously 0.23 is a bigger number. They're both decimals, but one's a really, 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 really small decimal. So on exponent scale, what I'm trying to say is that when you have a larger negative exponent, like 10 to the minus 6, that indicates a smaller and infers a smaller number. Okay, so these Ka values, as you go down the table, get smaller and smaller and smaller. And what we said is that the smaller the Ka value, the weaker the acid. So trichloroacetic acid or glutaric acid, which one is a weaker acid? Glutaric acid. Okay, so this one's actually the weakest carboxylic acid on the entire table. We know that because it has the smallest Ka value, or what's a lot easier when you know the trend is you can just look at the pKa values. A larger pKa value implies a weaker acid. Again, if this confuses you and you don't want to keep this straight in your memory, throw this on your note card. okay? So am I gonna ask you to calculate Ka on an exam? No. Would I give you a Ka and say, hey, what's the pKa? If I give you multiple pKa values and I say, this pKa belongs to this acid, this pKa belongs to this acid, tell me which of these is the weakest and which one's the strongest. Absolutely, I could do that. And from this table, I would want you to be able to order those for me. So if I said hexanoic acid, oxalic acid, and formic acid, we'll just say 1, 2, and 3. If I gave them those numbers and I said, which one is the weakest? Which one is the strongest? Could you put those in order for me? Which one is the weakest? Which one is the strongest? So remember, look at the pKa values, the associated pKa values. We got 3.74, 4.89. Oxalic acid, oh, that's confusing. Let's go with the larger pKa, we'll go with that one. So based on the 3 pKa values that I circled, I didn't realize oxalic acid has 2 before I circled it. Which one is the strongest and which one is the weakest? Could you put these in order? Anybody have a guess?
Is formic acid the strongest?
Formic acid will be the strongest. So we'll put it at the end. That's the strongest. Which one will be the weakest? Remember, the larger the pKa value, the weaker the acid. So which one has the largest? Hexanoic, the one I circled, or oxalic acid? Yep, Cameron said it, hexanoic. Okay, so that's number 3. Should be at the end here. 3 and then 2 and then 1. So it'd be hexanoic is the weakest, then oxalic acid, formic acid is the strongest. Now, I know that seems to not follow the trend as you go down the table, but again, always pay attention to the Ka values and order them that way, or the pK values, sorry. So if I give you a Ka and I say, hey, what is the pKa value? All you do is you grab your calculator, you punch in, don't forget the negative sign, negative log, and then you literally plug in the number for the Ka value. 6.5 times 10 to the minus 5, or whatever the Ka value is. That is how you calculate. You can double check yourself on this table if you want. That's how you calculate pKa values.
I have a quick question.
Yes, Abby.
Did you say, like, I was gonna put like the words on like that on the note card. Did you say we need to put that whole chart on the note card?
No, you don't need to put the whole chart. If you know these
Okay.
trends and you understand these trends, that should be enough to help you.
Okay, awesome. Thank you.
Yeah. All right. Last thing, I actually mentioned this. So we said carboxylic acid undergo, here it's saying neutralization reaction with strong bases to form carboxylic acid salts. So basically earlier I just said carboxylic acids can give away their hydrogen and they form the carboxylate ion, okay? And then we looked at an example when there's a main group 1 or 2 element around, they form those salts. We wrote out sodium acetate. That was an example of a carboxylic acid salt. So this is just the same thing in reaction form. It's an acid-base reaction by definition because this is the acid. The carboxylic acid obviously tells you in the name it's an acid. And we have sodium hydroxide here. This is just a reaction with some kind of base. We know acids and bases neutralize each other. Why? Because they make a neutral product. Water is always pH neutral. Okay, that's why they're termed neutralization reactions. Okay, so, and this is the exact same thing we wrote out earlier. So we got acetic acid and we got sodium hydroxide. The cation from the base is going to pair up with the anion from the acid. And that's where you get sodium acetate from. So this is the, I'm gonna highlight it. What we drew earlier was the carboxylate ion. It's the same thing. This is the, remember the name ending ATE? This is the carboxylate ion. But when it is paired up with a cation, we just call it by its ionic name. We actually call it an ionic compound. The reason you see the word salt here, like why am I calling this a carboxylic acid salt? Salt is just the common name for the combination of some cation and anion. We say they're ionic salts. So you have the carboxylate ion is acetate, and here it is paired up with the cation from the base, sodium. And then the this acidic hydrogen pairs up with the OH minus from the strong base, and that's where you get water, which is neutral, right? And so that is why we're calling this a neutralization reaction. This is just acid-base neutralization reaction. The salts tend to be more soluble than the acid itself because you've got these strong charges, and water is a very, very polar solvent, and it readily dissolves things like salt. I mean, you guys know this. If you had a glass of warm water and you dumped some actual sodium chloride or any kind of salt in there, it would readily dissolve, right? Same idea here. Okay, so based on that knowledge, we have these two acid-base reactions down here, and it just happens that our acids are carboxylic acids. So you write the products. What products would we get? If it helps you, you can think of these as double displacement reactions. The cation from one thing pairs up with the anion from the other compound. So this is the acid, and this is your acidic hydrogen. Sodium hydroxide, potassium hydroxide, this is the base. So remember the H and the OH, these are gonna pair up, and this is where you get water, the neutral product. So what is the other product gonna be? You don't have to name it for me, but at least be able to write it out from what you see. So we've got CH3, CH2, CH with a branched, this is in parentheses, so that methyl group right there is branched. And then COO minus. So if we draw this out, looks like this. This is the methyl group that's branched, and then we got the carbon, no carbon, and then O minus. It has given away its acidic hydrogen. And then it pairs up with sodium from the base. I'm not asking you to name this, I'm asking you to just draw the products. And then the other product would be what? So we've got our, yeah, thank you, Madison, water. Okay. B, we are given the name. So we have to unravel the name and draw this out before we can see what happens. 2,2-dimethyl pentanoic acid. So I'm gonna hone in on this last part really quick. I'll worry about the substituents later. Pentanoic acid. The root word there is pent, a 5-carbon carboxylic acid, with carbon number 1 being your carbonyl carbon. You draw that out? Pentanoic acid. So you should have 5 carbons. Four, 5. So this one is my carbonyl carbon. And I have OH. This is pentanoic acid. Now I have to add in the 2,2-dimethyl part. So if this is carbon 1, your carbonyl carbon, remember, is always carbon 1. So you've got a methyl group, 2 methyl groups off that second carbon. So this is carbon 2, So that means I'm gonna branch a methyl group up and a methyl group down, so it should look like this. And the rest of these are CH2. I'm gonna go back and fill in the hydrogens. So that is 2,2-dimethyl pentanoic acid, and it's gonna react with KOH. What products do you get?
Is there a reason that you have a line at the bottom of the C with the double line?
Oh, that's a number 1.
Oh, sorry.
That's all right. It looks like a bond, I know. It's a 1. Okay, so this hydrogen right here is the acidic hydrogen. It's always the one on the carboxyl group. That's the one that's gonna be eaten up by this OH hydroxide. And that's gonna make what? Well, we know we're gonna get water from that. The other part, we draw the same molecule and it'll be in its anion form, and it's gonna be attracted to the positive potassium cation from the base. Okay, so you will get this. This is the salt. I'm gonna put these in brackets. This is the salt. Our carboxylic acid salt. And then you always get water as the other product. Okay. All right. Any questions on that? So what we're gonna do, last things for today, we're gonna look at how do you make esters from we've been saying this whole time, esters are derivatives of carboxylic acids. But how do we make them? Okay, so substitution reactions occur with alcohols and amines. Yes, alcohols and amines. Yield water as a byproduct. So if you have a carboxylic acid and an alcohol, that's what we're going to look at first. A carboxylic acid and an alcohol. We're going to do a substitution reaction with a carboxylic acid as one reactant and an alcohol as the other reactant. This is how you form an ester. We call it, this is ester formation. It's known as, it's got this fancy word, esterification. I don't care if you know that word or not, but just know that this is how you form an ester. Here is kind of the general picture. Here's your carboxylic acid. How do I know it's a carboxylic acid? Right here. That's my carboxylic acid, my carboxylate group. Okay. And then the other reactant, if you look closely, this is an alcohol. Okay, that R group right here, this could be anything. It could be CH3. Okay, it could be methanol. It could be ethanol. It could be propanol, any kind of alcohol. Okay. What we're going to do, the parts that the text that are in red, these parts are going to break off and bind together and make water. And then the part in blue is going to bind with the remainder of our carboxylic acid compound, the part that is unchanged. That's what forms the ester. Okay. So this blue part, the OR portion, this is what is going to bind to that carbon. And that is where you get this ester from. Okay. Now you don't care about all my scribbles. Okay. So for example, if our alcohol is methanol, and then we just have simple O-acetic acid here, CH3, and then here's our carboxyl group, OH. What bonds break and what comes together? I'm going to draw this out slowly. This bond right here, that breaks. And this bond right here, that breaks. These portions, I'm going to trace them over in black. these portions, because they both break off, that is what forms water. You see that? HOH. And then once those are out of the way, this portion of the alcohol is just going to slide over and bond to that carbon. That's what forms the ester. Okay, so that's what all I just said is written for you right here in these bullet points. The OH or the carboxylic acid is simply, at the end of the day, is just replaced with the OR group from the alcohol. Okay, this requires a strong acid catalyst. That's what's indicated right here above these arrows, H+ catalyst. It gives you another example with butanoic acid and ethanol, okay? Again, just look closely at the red and the blue text. You see what breaks off and what comes together, and then what joins up to form your ester. So let's do a practice. You write the product of the following reaction. You have this carboxylic acid. It's some sort of substituted benzoic acid. And then you have methanol. I want you to write my products, or the products here. So should look something like this. We're gonna break this bond right here, and we're gonna break that bond right there between the O and the H on the alcohol. Okay, this is the alcohol. This is the carboxylic acid. So we should get this. Sorry if y'all can hear all that noise. It's annoying. Okay, so this is my carboxylic acid, and I have pulled off the OH. I'm going to highlight this part of the alcohol right here is what I'm going to now bond to this carbon. OCH3. It looks a little reversed, but that's the part that bonds. And that's what makes the ester, and then we know the other product is always water. Okay. A really, really good challenge problem. We're going to do B right here. We're going to work in reverse now. You're given the ester. I want you to break it apart and write the initial reactants, okay? Which carboxylic acid and which alcohol are needed to make the following esters? So you already have the product. Break it apart and write your initial reactants. I'll give you one hint. The bond that you need to break is right here, in case you're confused. And you're adding water back onto this. So when you break those apart, you should get this And then you should get this. Okay. So all I did was I broke this apart and I separated those molecules. Now water, if I draw water like this, HOH, okay. Actually, this part in blue is gonna go right there. This part, let's see if you can see green, is gonna go back onto the alcohol. So when we do that, we get OH there, and we just restore the alcohol. So this is the alcohol. We remade our alcohol and we remade our I'm gonna abbreviate, carboxylic acid. This is called a hydrolysis reaction. You see, anytime you take water and you break things apart and with water, we call that hydrolysis. Okay, I'm gonna skip over the raspberry oil problem because we need to see how we do this with amides. And then I'm gonna really quickly hit phosphate groups and then we're gonna be done. So just the same way that we form esters by taking an alcohol and a carboxylic acid, now we're gonna take an amine and a carboxylic acid, and we're gonna see how those make the amides that we've been naming and writing this whole time today. So carboxylic acid plus amines, that's what forms an amide. Here's what's gonna happen. The OH of the carboxylic acid is replaced this time with NH2. So very, very similar to what we just did with esters. Now we're doing it with amines to make amides. So here's what it looks like. Again, the parts in red break off, make water. The part in blue slides over and joins up with the remainder of your carboxyl group. And that's, this time it's a nitrogen group though. So that's what makes the amide. So there's several different examples here. The book gave like so many, because you can obviously have primary, tertiary, or secondary or tertiary amines. So we'll just look at the example here with a formation of a primary amide. You've got NH3. Anybody remember the name of NH3? We said this had a common name.
Was it ammonia?
Ammonia. Okay. So I'm going to draw ammonia out. Looks like that. So all we're going to do here is we're going to break one of these hydrogens off. And that OH and that hydrogen, that's what's going to give us water. HOH. So that's what's going to make water. And then the rest of this group, I'll box it in blue since it's typed in blue. That's what's going to slide over and bond to the carbonyl carbon. And that gives you, that forms the amide. When you're done here, we just have this one individual carbon nitrogen bond. So that is why we say we're formed a primary amide. And that happens if your starting material is ammonia and only ammonia. If your starting material is a nitrogen, so right here, I'm going to draw an arrow, is a nitrogen that has one alkyl group bonded to it, then what you will form, same type of reaction, what you will form though is a secondary amide, because what you end up with is this nitrogen will be bonded to the carbonyl carbon, and it was already bonded to the methyl group. So that's why we call it a secondary amide. Same reaction. You form water as well as a byproduct. If you start out with a secondary amine here, so this nitrogen is bonded to 2 methyl groups. And when we break off the H and the OH from the carboxylic acid, H from the amine, you make water. What you end up with, if we draw this out, is we've got this nitrogen is bonded to 1 of those methyl groups. I'm going to draw another arrow because we got 2 bonds to 2 different methyl groups, and we got another bond to the carbonyl carbon. So that's why we're saying this is a tertiary amide. Okay. But it's the same kind of reaction that we just looked at with esters. This time it's with an amine, so that what we form is our amide. We'll do more practice with this in the lab today, but I really would like to actually get to that now. So last thing, just like what I said, we did a practice problem 2 slides ago. We said, oh, here's our ester, break it apart and write the original products, okay? Like I said, we call this hydrolysis, okay? So I want to read this right up here for you. Reactions leading to the formation of esters and amides can be reversed, okay? So notice the reversible arrows right here. These are known as ester hydrolysis and amide hydrolysis. Anytime you take water and you use it to break apart molecules, we call that hydrolysis. What it does is it regenerates. So we're looking, here's our ester. Here's water. We regenerate the carboxylic acid and the alcohol. Or with amides, we'll regenerate the carboxylic acid and some kind of amine, okay? So esters, it's pretty straightforward. Again, you regenerate your carboxylic acid and you regenerate the alcohol. Amides are a little bit more difficult, okay? Amides are more stable in water. And so depending on what your environment is, you've got to heat it, you've got to add a lot of heat to break these apart. And you'll do hydrolysis with either an acid or acidic, an acidic or a basic condition to yield different products. So the simplest way I can explain this to you is with this figure right here, okay? If you do the hydrolysis with acidic conditions, what you're going to produce is a carboxylic acid and an amine salt. We looked at amine salts a whole lot yesterday. So I know you're thinking right now, okay, what is an amine salt? What is an amine salt? All that means is you have an ammonium nitrogen. Okay, remember ammonium is a nitrogen with 4 bonds and a plus charge. That doesn't mean it's a quaternary amine. Not all those bonds are necessarily to carbon, but you're forming an ammonium ion. Okay, so walk through this with me. Here is our amide. We're going to hydrolyze this, but we're going to do it under acidic conditions. That is indicated to you right here. Acidic conditions, H3O+. We're going to heat this up. And what you will form is the carboxylic acid. That's right here. I'm going to highlight the carboxylic acid. Okay, there's the carboxylic acid. We remake that. But we also make an amine salt. This is the amine salt. If you draw this out, N, H, 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 and we got a methyl group, CH3. And this nitrogen, because it's got 4 bonds, has a plus charge. That is what we're calling the amine salt. So if you try to hydrolyze an amide under acidic conditions, you form an amine salt. Okay. If you do it under basic conditions, you produce carboxylate anion and a neutral amine. Okay, so here is our, so this one is acidic conditions. This one is under basic conditions. Here is again the amide. How do I know it's an amide? This part right here, that's the amide. You see the carbonyl carbon and you see the nitrogen group branched off it. This right here tells me OH minus, that's a base. So I'm hydrolyzing this under basic conditions. My products are the carboxylate anion, and this is the free amine. Whoops. Oh, sorry, it had it labeled over here for you anyways, acidic acid hydrolysis and base hydrolysis. I double labeled it for you. So amides are complicated, way more complicated than esters. Maggie, you have a question.
Yeah, okay, so we labeled the acid and the base and those two right there. But we only labeled in the first one the acid. So does that make that like automatically means that the amide is the base? Does that make sense? Because like everything in equations we've been labeling an acid and then the base. So that would automatically mean that the amide was the base.
So if this is your, well, here's the deal. This is not just I'm not going to ask you to label one as an acid or a base. This is not a neutralization reaction. What we're doing is we're breaking a molecule apart. We're not just transferring a hydrogen here. So I am not going to ask you to say, which one's the acid, which one's the base. This is a hydrolysis reaction, not a neutralization reaction, okay? I don't know if that helps. So you are right. When we talk about acid-base stuff, that's a neutralization reaction. I do have you label sometimes the acid and the base to help you see how that reaction is going to go. This is different. Here, we are taking this whole molecule right here and we're breaking it into two pieces. And where are we breaking it? Right here. Right here. We're breaking that bond. And we're making some kind of an amine. It might be a free amine or an amine salt. And then we're restoring some form of the carboxylic acid. It might be the neutral carboxylic acid. Like up here, if you have it, that's terrible, Henry. T-U-R-A. It might be the neutral carboxylic acid, or it might be the carboxylate anion, depending if you do this under acidic or basic conditions. Okay, that gets us through formations of esters and amides, and then the reverse of that, which is hydrolysis of esters and amides. That might be the one biggest takeaway I want you to write down. So we talked about formation of esters and amides, and then we did hydrolysis. And these are opposites, okay? Hydrolysis is the opposite of the formation. One forms the ester amide, one splits it apart. If that helps you to kind of summarize it. There we go. Okay, lastly, I'm just going to introduce the phosphate group to you. I'm probably not going to really get into everything these slides entail. I know we're tired. I'm tired of hearing me talk, so I know you are. Lastly, phosphoric acid is similar to carboxylic acids, but phosphoric acid is different. Carboxylic acids have one acidic hydrogen, just one. Phosphoric acids have 3 acidic hydrogens, and it can lose all 3. So here is a comparison between carboxylic acid and phosphoric acid, and the 3 acidic hydrogens are in red. And if you follow this figure through towards the right, you see that phosphate group losing 1 hydrogen at a time. And you see the different derivatives. You got a dihydrogen phosphate ion, dihydrogen, because we got 2 hydrogens left. We lost 1 hydrogen, so now we just have a -1 charge. Lose another hydrogen, and we just get hydrogen phosphate. And now we got a -2 charge overall, 1 hydrogen left. When we lose the final hydrogen, we got the phosphate ion with a -3. This was a polyatomic ion that maybe you memorized in a previous class. Phosphate ion. Okay, why is phosphate important? Can anybody think of a single molecule that might be important that would have phosphate involved? There's 2 big hitters in our bodies, ATP and DNA. Both largely depend on phosphate, okay? Last thing I want you to know from this slide. Phosphate groups, they can be added to organic molecules. So we can phosphorylate something, or they can be ripped off of organic molecules. We can dephosphorylate something. This is showing you an organic molecule. I don't care if you know what the organic molecule is. It's a carboxylic acid, but we can put a phosphate group on it, okay? That's called phosphorylation. We'll phosphorylate that. I don't care if you know, you don't have to know that it's called a monophosphate. And then at physiological pH, these phosphate groups are in their ionized forms. I do want you to know that. At physiological pH, these phosphate groups are in their ionized form. What that means is they have some kind of charge. So in fact, if you just wanted to write at pH 7.4, this is the form that they're in. They're usually charged, -2 charge. Okay. I didn't even go over this, but why are we calling it phosphoric acid? You might have noticed instead of a carbon in the middle, it's a phosphorus in the middle. I'm sorry, I should have said that at the beginning. You probably noticed that though. Okay. Okay, last thing. This seriously is the last slide, then I promise we're done. Phosphate groups can join together to form diphosphates and triphosphates. Okay, this is where we're getting into molecules like ATP. ATP itself is an adenosine base. I'm not going to draw that out. You don't have to know what that is right now. But look, it's got 3 phosphate groups. 1, 2, they kind of overlap. 3. They're joined together by these little ester bonds. 3 phosphate groups. So that is why we call it adenosine triphosphate. The tri there indicates that there's 3 phosphate groups bonded on to this organic molecule, adenosine.
Why is there? Oh, sorry.
Go ahead, Maggie.
Never mind. That's a really dumb question.
You're good. So with ATP, here's what's really cool. For some reason, so I'm going to erase my boxes for a second. For some reason in nature, when we break apart, I forget if it's this bond or this bond, but one of those bonds, and we'll see which one it is later. When we break apart one of those bonds, it releases a ton of energy in our bodies. And our bodies are smart, and they harness that energy to drive other reactions, okay? So anybody, when you're running a fever, you can feel your forehead, it feels warm. But go ahead and touch your forehead right now. Does it feel slightly warm? Seriously, do you feel kind of warm? You are one giant exothermic reaction. Your body is releasing energy all the time. So part of the energy released from this is just to maintain body temperature, okay? Just to maintain our stable body temperature. But there's something really, really special about breaking these phosphate bonds that releases energy, and our body uses that to drive other reactions to do all the things that it needs to do. So this is a really, really important thing. We're going to spend a lot of time on this when we get to oxidative phosphorylation. And that's it. We're done. Everybody say, praise God.
So we don't need to know the carbonyl group substitution reactions?
No.
So not on test, mark it out.
I mean, this is kind of a review of, this is kind of just a review of what we looked at.
Okay.
So yeah, just don't worry about that. Just focus on what we've already talked about. I may regret not going over that, but I'm just like so done. Okay. Let's just take a breather for a second. If you need to go to the bathroom, if you need to get a snack or whatever, I'm gonna slowly pull up the lab